Okay.
Hello, everyone. Welcome to the Geoscience and Geoenergy webinar of 13th of January 2022. This is our very first webinar in the new year. So happy new year to all. With best wishes to all of you, we are today delighted to kick off the webinar of 2022 by hosting our dear friend and colleague, Dr. Nicol Dopffel from NORCE on a very special topic. Actually, we have never hosted anyone in this topic, reservoir microbiology. So Nicol, you are going to educate us and train us on this subject as well. To those of you who don't know yet already, Nicole has studied biology at the university in Tübingen in Germany. During her PhD, she specialized on uh, anaerobic microbiology with the general topic of microbial iron oxidation and iron reduction. From 2014 to 19, she worked as lab team leader and research scientist at the R&D department of the chemical company BASF in Germany, where she mainly worked in oil field related topics and microbial contamination problems in the industrial sector. In 2019, she moved to the Norwegian Research Center NORCE in Bergen, Norway, focusing on different aspects of microbial effects in the energy sector. It's our pleasure and honor to host you, Nicole, today. Thank you for graciously accepting our invitation. And thank you for kicking off the year of 2022 series of webinars that we have. To the audience, please note, like always, it's a new year, but we stay having the same webinar as always. The talk will last for about half an hour, followed by questions and discussions. Like always, please do text your questions in the chat box after the lecture of Nicole. Sebastian would share the discussion session. Do not wait until the end of the talk to ask your questions and type them. Please do type in your question anytime you feel appropriate. Your question will motivate many others to also engage with the speaker and present and ask their question. So for a second, we have lost our speaker. We are going to wait a bit until she is back again with the same link of the broadcast. So you should be with us. Sebastian, I've heard that you are a good singer.
No, Harry, we have done this before. We have threatened our audience with us singing before. I think that's the last thing we want. So hopefully, for the sake of our audience, hopefully Nicole is coming back in a moment.
Yeah, I'm sure she will be back in a moment. Are you sure you're not going to sing for us anything? You could sing.
No. I'm just going to send Nicole the link again,
yes,
just in case.
Exactly. That should be fine. No, she was with us, but just suddenly got disconnected. So she will be back in a second, I'm sure.
We had, how many webinars did we have so far? Roughly
88.
88.
I guess this is our 88th speaker,
88,
the first time. Yes.
The first time we've lost a speaker online.
Yes.
So.
But that's okay. I mean, she's texted that she'll be back in a second. So
she just texted you?
Yes, she did.
Okay, fantastic. So.
Yeah, just the laptop, I guess, just crashed. It should be fine. So we are starting the year with a little bit technical difficulty. That's fine. I see many people are joining in, so that's good. So if you have just recently joined, the speaker laptop just crashed, and we are waiting for a few seconds until she comes back again online and delivers her talk. So stay with us. Sebastian is not going to sing for us. I'm not sure if it's a good news or
I
or
think
bad
that's
news.
fun. That's absolutely fantastic news for everyone. There are a few things you don't want to listen to, and that one of it is me singing.
So, okay, so we are gonna start very soon. As soon as the speaker is back, just bear with us for a few more seconds.
I'm also just going to put in the chat that
yes, exactly, that we are just waiting for the speaker to reconnect. So Happy New Year, everyone who is online. We are waiting for the speaker who just got disconnected suddenly, and she will be back in a second. Stay with us, please. And you can also use chat box to say Happy New Year to your colleagues and friends as well. So I have Mohammed Rawahi saying Happy New Year to all. This is great.
there's still no sign from
No,
Ansaida's
she
there.
says, yes, she says that she is having a network problem. Hopefully she'll be back.
Very nice to see you back online, Saida. Saida was one of my PhD students.
Oh, okay. Very nice. If you have just joined, I see many more people are joining in. We just lost our speaker due to network problem, and we are waiting until she comes back online again. So stay with us, please, and we hope.
Adi, one of the things we could actually do while we're waiting for Nicol to come is just, I'm going to do this, get this back up here in the background, is to give an update on the speakers for
absolutely.
next few
yes, weeks. Absolutely
weeks.
right. Yes.
So we do have Nicol speaking today as soon as she's back, and then next week we have Catriona Reynolds from drugs. She's an expert in BECS, and she's going to talk about some of the social policy around energy transition. We have Ian Main speaking on the 27th of January. Ian Main is from Edinburgh University. He's talking about catastrophic failure of earth materials, so earthquakes, etc. And then we do have a change in program on the 3rd of February 3rd. Felipe Pereira, who was originally scheduled, had to decline due to family reasons, and he is being replaced by Auke Barhorn, one of Hadi's colleagues from the Technical University of Delft. And I assume is also going to speak about deformation of rocks. He's an experimentalist. And we see Nicol is slowly coming back here.
Nicole, hello, do you hear us?
Yes, I can hear you. Can you hear me?
I do hear you very well, but we do not see you. But that's okay, because as far as you,
I think I have to. My network is a little bit
okay,
slow, so
okay.
I think I need to keep the camera off. I'm very sorry.
No problem. That's absolutely fine, at least for the first half an hour of the lecture. So are you going, would you please share your screen with us? And
Yes.
we hope that we can see your screen.
Yeah. So I'm very sorry. It's of course everything is running, and then
that's okay.
It's
the start of the new year, so that's okay. So
yeah, you probably had some other introductory words, I hope.
Okay, I'm going to add your screen to this. We see your screen perfect. So would you please press a presentation?
Yes.
Excellent. So everything is perfectly set. So before we lose you, we start the session. So everybody, welcome again. Nicole will give a talk about subsurface reservoir microbiology, relevant for energy sector. And then after about half an hour, we will hold the discussion session and question and answering that Sebastian will chair. Nicole, the stage is all yours.
Perfect. Thank you very much for the introduction. I'm very sorry about the technical difficulties. It's always in the worst moments. But let's get started. I'm very excited to be here. Thank you very much for the invitation to talk about this really, really cool topic, and that's subsurface microbiology. So, and I want to make also really some introduction into the topic at all, because I think we have quite a different audience from a scientific background. So I thought it would be good to have some introduction here. So when I talk about microorganisms, what do I actually mean with this? So of course, microorganisms just means it's a small organism. So just unicellular organisms. But then again, what does that mean? So you can see here a very old tree of life, which was published in 1990. And you can see that from our last universal common ancestor, there are three different big groups of life. That's the bacteria. I think everybody heard of that. And if you look under the microscope, you will see a small round shape. But then there's also two other kingdoms of life, and that's the archaea, which came also from our common ancestor. And then we have the eukaryotes. So when you have the archaea, they are also singular cell organisms. If you look under the microscopes, again, you will see something small and round. And then from the eukaryotes, that's actually also us. So we are here in the animal kingdom. There's also the fungi and the plants and some others. And of course, there you also have, for example, yeast. If you look under the microscope, you will have something round and small, but a little bit bigger than the other ones. So you can already see things are always quite small. But this tree of life is now very old, and I want to show you a more recent one, published in Nature 2016. And you can see that actually it's a little bit more complicated. So in the top, you see the kingdom of bacteria based on DNA sequences, which were sampled all over the world. And you can already see that it's huge. And then down here, you can see the archaea, and then you can also see then the eukaryotes again, where we are. And also the difference in length also tells you something about diversity. So you already can imagine the huge diversity there is already just within the kingdom of bacteria. So, but where do we actually find microbial life? So that's actually, you can say, almost everywhere. That's you can find it in soil, sediments, very extreme environments like this hot spring, of course also in oil and gas reservoirs and subsurface aquifers. And as a very rough rule, you can say if the conditions are more extreme, you will find more archaea, and if the conditions are more ambient, you will probably find more bacteria. But this is really just a rough rule. And then you have different factors which can shape your microbial community, which you will find. And the most important one is for sure the temperature. So you can say we have a hard limit of life at 122 degrees. And you can go low to -20, but at least as long as you have liquid water, you will find microbial life. And here I just want to add, because maybe now a reservoir engineer somewhere says, all my reservoirs are over 120 degrees. Perfect, I have no problems. I will now stop listening. But here I always want to say that it is really important that you consider cooling effects in your reservoir. Because I had it once or twice that people came to me and said, my reservoir is over 90 degrees, there shouldn't be so much activity. But then, of course, after decades of water flooding with cold water, the temperature in the near wellbore area is not as it was when you started the reservoir. So always consider cooling effects, and these can be quite important for the community down in the subsurface. Then other factors, of course, pH. pH 7, you will find most of it, but you can find specialists to pH 11 or 12 and down to pH 0 and 1. Then salinity, of course. Again, if you have high salinity, you will get more specialized communities, but you can go as high as saturation. They are actually living in salt lakes, so the salinity is not even this as a big factor. You have different metal concentrations, of course. Pressure seems to be a community factor, but very unsure. There has not been much research on this. And then, which is important for the subsurface, permeability. If you go really, really low in the permeability, then the microbes just don't have space to live there. So if you really go in the low millidarcy area, then it will get difficult. So as a rough idea for you guys, a microbe is around 1 micrometer in size, very rough. So if you go really low in your permeability, then you could get actually sterile conditions. Yeah, and then of course, different nutrients shape different communities. So, and for the subsurface, there's always this question. So how deep can life actually go? And there was this really nice expedition in 2012 in the coast before Japan, where they drilled almost 2,500 meters below seafloor, just to look if they still find microbes. And they did, actually. The cells were there and they were active. And when you consider the amount of cells, which was actually not that high, it's 10 to the power of 4 cells per cubic centimeter, is not much. But if you consider all the subsurface, you get to this huge number of cells, which actually accounts for almost 20% of all microbial biomass we have on the planet. So until now, there is no depth limit found, at least when it comes to pressure. Of course, if you go too deep and the temperature goes over this magical 122 degrees, again, there will be no life, but this is not related to death. So there's actually still a lot to discover, and it's a very, very big and unknown environment for us. So what do they do in the subsurface? So what can they do? So I just want to show you what metabolic activity microbes can have. So we start, of course, again, what kind of nutrients do we have? We have organic matter, which can be hydrolyzed into, for example, sugars, lipids, amino acids, which can be fermented, again, by microbes. And then you can have, for example, acetate, hydrogen, and CO2. And then you have a lot of different possibilities. So you start, like us, with oxygen respiration. And then when the oxygen is not there, and there's a lot of environments where you don't have oxygen, you start with nitrate reduction, then you come to manganese reduction, iron reduction, and then at the bottom, you get sulfate reduction and methane formation. So this redox cascade, this is now a very simplified view of it. just depicts what will happen when you have different electron acceptors present. And you can already see down here one of our friends, the hydrogen, which I will talk a little bit more in detail afterwards. And all of these are, of course, cycles. So you will get, for example, H2S, which then can again be further metabolized back to sulfate, for example, or to sulfur. So there's a huge variety going on in the subsurface. As I say, it's always much more complicated than you think. So let's get a little bit more into the topic of the energy system. So now you saw, okay, microbes, they are everywhere, they can do a lot, but what does that actually now mean for us when we talk about energy systems, for example, when you're engineering something? So here you can see an overview of different energy systems, for example, oil production. So you have top side, you have a salt cavern, you have the reservoir, aquifers, geothermal installations, of course, wind turbines. So there's quite a lot where humans are actually designing energy systems. And then there can be positive aspects of microbes. For example, they can help with bioremediation in case of an oil spill. You can have biorefining or biogas production. I think this is very common in many countries now. But most of the time, the microbes can actually cause problems. So you can see here now a lot of red dots. So you can have microbial influenced corrosion or material corrosion, souring. As I showed you, they can produce H2S. You can have storage of fuel fouling or just plugging and clogging of your systems. And here on the side, you see some pictures what microbes actually do. So on the top, you can see microbial corrosion at its work. So these microbes actually corrode steel pipelines. You can have just biofilm clogging of your pipelines. And this down here is a very extreme example of a groundwater well in Australia. So, but then again, so what about the hydrogen? So I want to talk a little bit more into detail that. So because there is now this topic of hydrogen underground storage, which is now coming up because hydrogen will be an important energy carrier in the future, but where to store it? So we need the space, and here different storage options are under discussion. For example, gas reservoirs, salt caverns, or deep aquifers. And then, as you just saw in my previous slide, these are all living spaces for microbes. And so it actually, these systems are not sterile. So when you design them, you have to think about them also. Because hydrogen is a perfect electron donor for a lot of different microbes. So you can see on the side, actually, a little bit what I showed you before, like sulfide reduction, iron reduction, everything can be fueled also by hydrogen. So it really just depends what kind of acceptors you have and what kind of hydrogen concentration the hydrogen will be metabolized. That's also a reason why you will not find very high hydrogen concentrations just in soil, because it will be metabolized so quickly. So there's actually competition going on the hydrogen between the microbes, which also means you can have multiple processes on the same time happening. And that means also for our underground storage that you will have direct contact between the microbes and the hydrogen. And then the question is, what will happen? So for example, again, the souring H2S, what I mentioned before, I think you heard about it. So that means actually that sulfate is reduced to H2S. There are a lot of microbes which can do that. You can identify them very often because they have the disulfo in the name. They are active in most anoxic environments, and they can produce a lot of H2S very fast. So there's always some, even from low sulfate concentrations, you can get a lot of H2S. So here you have a major risk. If you have hydrogen, you can actually have potential souring, and of course the consumption of your hydrogen. And then another one I want to highlight a little bit is the microbial influence corrosion, because I think that is something which is maybe not so known. So that means that microbes can actually corrode steel and iron. It's a very complex interplay. I don't want to go into too much detail because you can make a whole own presentation just about MIC. But I like to show always this picture from a paper where you see they had this key with a sulfide reducer, and after 9 months and after cleaning, this was left of the key. So these microbes actually, you can say they eat the steel, they eat the iron. So if you're interested, there's actually a cost action plan now starting on this topic. So because this process is also fueled with hydrogen, so it's not clear how important it will be for the topside installations or also the subsurface installations, but it could cause significant damage. So something to be aware of for sure. So what I want to do now is a little bit to highlight the aspects between a salt cavern and a porous media. Because these are the most common ideas of hydrogen underground storage. So in an artificially leached cavern, you will have initially quite a high cell input, but you will also have quite extreme salt conditions. But that, as I hopefully convey to you, high salt conditions doesn't mean no microbes. So you will have probably experts in these salt caverns, and you will have regular input of fresh hydrogen. So it will be quite interesting to see what you will find in these kind of caverns and what will actually happen to the hydrogen. And then you have the porous structures, which are, they always contain very diverse communities, at least if you again are in the range of life. And these communities are always different depending on your reservoir. So no reservoir is comparable to the next. You always have a different community, at least that's my experience. But you have longer storage cycles, larger surface area. So it's again a lot of interesting questions arising when you would input hydrogen into these porous structures with microbes. What will happen? Then, of course, hydrogen underground storage is not like an idea which just came up. It is something which has been done for quite some time. And there are now a lot of research projects ongoing, which is very exciting. So all over Europe and also outside of Europe. And you can just see here a table of the most well-known underground storage sites. A lot of salt caverns, which are in industrial use, and then also some porous media. Very famous is the one in the Czech Republic and Lobodice, where actually the first time microbes were observed. Because you can see here in the green, all these actually experienced microbial activity and hydrogen consumption. So they injected hydrogen and they were not able to get all the hydrogen back. Here I want to now show a very nice experiment from a working group at the BGR in Hannover, Germany. So this data is nicely supplied by Anja Dormann and Martin Krüger. So they took water from porous media, put them in a bottle with hydrogen in the headspace, and then looked what happened. And you can already see that every time they put some hydrogen into the bottle, it got consumed pretty fast over time, and again and again and again. So these microbes can actually feed on this hydrogen quite efficiently. So you can already imagine, and when you inject your hydrogen into your subsurface, that you will not get it back. And then again, I want to show some very nice data from the Underground Sun project, which was done in Austria. You can find all the data in the published end report on their website. And it's they again took the water from the porous media, got it in contact with hydrogen, and you see the hydrogen is consumed over time. And on the same time, they got actually methane production. This was now in the lab, and they observed the similar thing also in the field. Of course, still some questions remaining, as always, but you can see the blue line, hydrogen goes in, and then hydrogen coming out, and there was actually some missing. Again, could be also a point of diffusion, but there has been quite strong hints that microbes were active. That was also seen, for example, when they looked at the RNA, so the activity of the microbes, they observed methanogens in their reservoir. That means methanogens, like you remember from the beginning, they use the hydrogen, they use CO2, and then they produce methane. But this actually got to a new idea, and that was actually the Underground Sun conversion project. So this is called biomethanation. There are also now more and more projects on it, and it's actually quite a neat idea. So it means that you could use CO2 from the industry, for example. You can use hydrogen, which you produce via electricity, pump it into your reservoir, and then the methanogens will convert it to methane. And then you can use the methane as a natural gas, uh, like we do already. So it would be actually a very good concept, and it would be also a CO2 sink. As long as, of course, as you capture the CO2, this is important. And here I also want to highlight, because there was just recently before Christmas, there was this really nice Nature article where they actually observed this process also in a CO2 storage site. So this was in the US, where they injected CO2 in a hydrocarbon reservoir and they got methane. Here, the fascinating thing is they did not inject hydrogen. The hydrogen was produced in situ from the hydrocarbons. And that is actually really, really cool. And I want to also show you that microbes, they can also produce hydrogen. It's not only that they are our enemies, they can actually also be our friends, because these so-called dark fermentation is very widespread. They can use a lot of different sea sources, for example, hydrocarbons, but also glucose or waste, and you get hydrogen out. And they're different organisms. So you can actually also see them that this could be an option for producing hydrogen. And there are ideas to use maybe some organic wastes, use these microbes, and then you get hydrogen and you can fuel your car. So that would be an idea, your waste container at home with the microbes producing the hydrogen. So there are always different sides to look at it. Yeah, and then at the end, I just want to say, okay, you're now interested in hydrogen storage, but you are like, okay, do we have microbes? What do we do? So how to start? Of course, it's always depends on your project, but you can see here this very simplified line. It's sampling, transport, analysis, which generates hopefully knowledge. And at the end, there's the big question mark, can we actually mitigate it? Because of course, we want to also solve the problem. Not only say, okay, we have microbes, but then what do we do? How can we actually deal with this microbial hydrogen consumption? So there are a lot of different ways, of course, to analyze it. I just want to mention a few. Of course, there are papers around. You can have a look. Different methods, for example, very important, the DNA community analysis. So you need to identify them, you need to count them. Still, I think cultivation is very important. Of course, not everything can grow in the lab. This will always be the case, but if we can enrich them and isolate them, we will also gain a lot of knowledge about these specialized microbes. And then we can identify growth triggers and maybe inhibit them. That would be the idea at the end. Of course, analytics, very important to know what we're dealing with. Again, temperature, pH, salinity, the major community shaping factors. But also it's always important to know what kind of electron acceptors do we have, what kind of organic compounds, do we see gas changes, and also minerals can play a role. I didn't go into detail on this now, but of course this is also everything which is present has a role in the subsurface. Yeah, so many open research questions, which I hope will now in the next years will be resolved or at least started. So what type of microbial types do we have, which are crucial? Maybe we'll find always the same, but maybe they will be all different. Maybe each salt cavern will be a different world of its own. What are the microbial rates? This is very important for modeling and risk analysis. Pressure effects, maybe there are thresholds, maybe this can actually be used as an inhibition or not. Is the inhibition at all possible? Then the geochemical effects. And then again, another idea would be maybe you can use these microbes also for leakage detection if you actually know who is triggered. So a lot of fascinating research, and I want to use my last slide actually just to really shout out that for these kind of projects, we need different sciences. We need cross-disciplinary projects because we need sometimes reservoir engineers and microbiologists, they have different languages, and we can actually sometimes we need to understand each other, and the microbial data is important for many different things like the modeling, the safety, the material selection, the storage selection, the monitoring. So it actually will be really, really crucial to understand it and then be able to work with it. So we need cooperation between the sciences, we need cooperation between academia and industry, to really build now this basic knowledge and also field-specific knowledge. And with this, we can then be able to understand these very interesting types of microbes in these systems. So at the end, I hope I could show you that actually this microbial activity in the reactions, they are important. They can be positive, they can be negative for a lot of energy systems, but we should not be ignoring them. That's for sure. Yes, and with this, I want to thank you, and I'm open for questions.
Thank you very much, Nicole, for this extremely interesting lecture. Is it possible for you to now unshare your screen and try to turn on your camera instead and see if the bandwidth would be enough?
I will do that. Yeah, I will stop sharing.
Yes, that might help.
Yeah.
A lot, so that we see you, and it would give more chemistry, obviously, still, but your voice is pretty sharp, and so it would do well.
That's good.
Yeah.
At least that's good.
Or we
Yeah.
can find an avatar to place in your icon. A special microbe, for example, that is your most favorite ones. So thanks a lot. I see plenty questions are being posted. I hope that you could manage to turn on your camera midway. We just suddenly saw half of your face, but anyhow, otherwise we just proceed
Yeah.
with the questions. There are plenty questions posted, and it was an excellent ending to say that we in the reservoir engineering sector we need more and more collaboration and joint work with microbiologists, and you and I have been also discussing for a long time about joint works on this subject. So
Yeah.
with that, I'd like to give the floor to Sebastian to pick up the questions for you.
Yes, thank you very much, Nicole, for a really nice talk, and especially a show to the geoscience and reservoir engineering community to think about more about microbes and the impact they have, and do more collaborative work. And many questions, and trying to group them. So Yuan Wang kicked off with the first question and come up with a follow-up question later on. He says, "Hi Nicole, great talk. Can we control bacterial activities in the subsurface? So do we need to take their living characteristics as a given?" And he qualifies then this question later on, said, could you comment what we can do to prevent consumption of hydrogen by microbial activities? Because you made the point, sometimes they're bad, sometimes they're actually good for the subsurface. So can we engineer the microbes, the behavior of microbes, or do we just live with them?
Yeah, that's a very good question. I mean, that's always the dream, of course, to have at the end, can you shape the microbial community in a way you want? It's difficult, I have to say. Of course, I mean, like when you say, can we engineer them like genetically? Of course not. That will never work. At least not for the subsurface. And you should not release genetically modified microbes anyway. So from my experience, there are, of course, ways to move a community. For example, which has been done for the oil and gas industry a lot, you can inhibit souring by injecting nitrate. So, and if you remember the redox cascade, so nitrate stands above the sulfate, so you will actually inhibit the sulfate reducers, and then you by feeding the nitrite reducers. So that's a way, but you always have to be careful because there's no promise that it will work. So, and it has been, it worked once, sometimes it didn't work, or it bounced back, or there were actually then microbes growing both on sulfite and nitrate simultaneously. So it's difficult, I would say. So, yeah.
Thank you. So sticking on the topic that we sort of need to live with the microbes in the subsurface, Marty Bone asked, do you believe the reservoir's microbial activity is an important factor for site selection? So normally we look at permeability and volumetric storage and vicinity to the surface facilities. Do we need to consider the microbial activity in the reservoir as well?
I think so, yes. So I think when you screen for a site, that you should actually already look also at the microbes, that the microbiology should be one factor besides permeability, salinity, temperature, and so on, that you consider also the microbes. But because I think, I mean, it will show in the next years what will happen with the underground storage sites. But it could be actually that if you have certain microbes present or microbes at all, that maybe it will not work at all. So I think, especially now in the beginning, microbiology should be considered for storage site selection, at least until we know more. Yeah.
You made the comment that there are quite a number of questions around experiments and how unique are certain microbial communities. In your closing remarks, one was around, I'm trying again to group some of these questions. So
Yeah.
Farah said, no, are there some most common strains in salt caverns that consume hydrogen? You made actually the point and said that's an open research question. So again, I pick Marcia's comment here question, and she says, I believe it's very difficult to determine which microbial communities are present in the reservoir, which would make designing representative experiments difficult. So how do we actually tackle that challenge that we don't know how unique or different microbial communities are in certain reservoirs and how they behave, consume hydrogen, for example? How do we tackle that experimentally in a
Yeah,
smart way?
yeah, it's a good point. It's always like, okay, we don't know much. How do we start? It's I think it's right now it's good to start basic. Like, okay, we first need maybe some subsurface strains which are available, just to do lab experiments and say, okay, we get these kind of hydrogen consumption rates. This is at least a start. You can start calculating a little bit. But then again, this is not the field. It's not the reality. So crucial is actually using original samples from the site, which are tough to get, I know, but at the end, the samples from the salt caverns and the samples from the reservoirs, the fluid samples, they will need to be analyzed. What kind of community do we find? What kind of microbes do we find? And also used for experiments in the lab. And this will give us the first knowledge already. So what kind of community do we have? How many cells? And then this will again make things much easier in the future when you try to select for representative strains again. And this is then again we need a combination of academia and industry, because we need the field access. We need to really look at the real field sites, because they matter at the end.
Okay. Thank you. I'm trying to pick a lot of really interesting questions that colleagues, the audiences are asking here. So some of the more fundamental questions that are coming up, and Alice, a very interesting presentation. Do we expect microbial communities to evolve fast enough to eventually have different characteristics than when we started? What's the time scale of their evolution?
Yeah, that's an interesting question. So of course it depends a little bit. It's hard to say. So you will get always some adaptation over time. I really can't give you now a number, like it will take a month. I can't say it. There's so many influencing factors. Maybe they will take years, but then again, at the end, we want to use these field sites for decades. So it's really hard to say. I would say always you can expect that something will adapt or evolve over time. Also, or maybe it will be introduced. That's also another thing when we, of course, drill wells and flush things with seawater, for example. You will introduce a lot of other microbes, and then there will be competition going on and new strains which has not been there before. So it's a very complex environment then going on. But it's hard to say, like to really say a number. So this will take this amount of time. It probably also depends on the field site again.
But if I understand you correctly, so it is something that we could see changes to the microbial communities over the time span reservoirs being managed.
Yeah, of course. That's for sure. There are several examples. I mean, from the oil and gas industry, there is quite a lot of things known that, for example, when you start flushing with a certain chemical, and then after again, maybe months or years, you can actually have degraders of this certain chemical suddenly. And then, for example, or a polymer degrader suddenly appearing. And something which worked then very nicely before suddenly doesn't work, because then you have this threshold, you have the microbe coming up, eating your biocide or your polymer, and then it's not working again. So this is for sure happening. Yeah.
Okay, thank you. There's a question from Taufik Nassan. He also said, thank you very much. You mentioned the maximum temperature is 122 degrees centigrade. Does this apply to atmospheric pressure? Is it independent of pressure? And where does this number come from? So a couple of very fundamental questions.
Yeah. That's so this is now at atmospheric pressure, this temperature. Where does it come from? It's just it's the field site where we still found microbes living. So it was, I think, hot. Oh, what is now? I'm missing the English word. That a hot smoker, I think, in the sea, like these
The black
hot,
smokers.
yeah, black smokers, these
The black
hydrothermal vents. I think there they found it. So these are natural hot environments where microbiologists went and actually still found microbes living. So this is the number. And nobody ever isolated a microbe which could deal with more than 122, which then again, maybe we just haven't found it yet. So I wouldn't rule out that maybe at some point we will find a microbe which can have 123 or 24. Who knows? So that's it. And then, yeah, the effect of pressure, very interesting. There's really not so much known what pressure actually, how it shapes the community. There, a little bit it is known what pressure does to the membrane. So it's a little bit comparable to salinity effects. So the membrane and the proteins need to adapt to the pressure. But surprisingly, if you change the pressure very slowly, most of the microbes can actually deal with quite a lot. So they don't seem to mind pressure that much. But then again, pressure experiments are hard to do and not many people do it. So I think there's still quite some basic research to be done on this topic.
Okay, that's really interesting. Thank you. So lots and lots to work on. I'm going to do one more fundamental question, then we come back to a lot of questions on the hydrogen alteration. Sarah says, thank you for the interesting talk. I'm interested in the microbial tree of life. In the image, it seemed that bacteria proliferated better than archaea and eukaryotes. Is this correct? Why has that happened?
Ooh, so you can say that the bacteria, they are very, very diverse. As you saw in the tree of life, one factor is bacteria. They are not so complex, for example, like plants or animals. So if you have a mutation going on, you can have very fast a new substrain of microbes. They also have very efficient ways to exchange DNA, even between species, between genera. So there's a lot of DNA exchange, there's mutations going on. This comes again back to they can adapt fast and slow. And then this makes this huge diversity. And then every environment gives rise to new microbes. And there are very complex synergies between microbes. And so this, I think, explains why there's such a huge diversity. Why then the archaea are relatively small? I think, yeah, probably because the bacteria occupy a lot of space where the archaea could be. So the archaea are more into the more extreme environments, and there are less extreme environments than more the ambient ones, like the open sea or the soil. Yeah, and then the eukaryotes, like we are very complex. So for very complex animals and organisms, we evolve just more slowly. That's the reason. So until from us, like humans, to come something new, it will take a few years, and bacteria don't have that problem.
Okay, great. Thank you very much. Going back to hydrogen interacting with microbes interacting with hydrogen, Farah says, microbes are known to prefer the consumption of dissolved hydrogen over gaseous hydrogen.
Yeah.
How much quantitative evidence is there of biological alteration of hydrogen at a storage site?
How much is it? I don't know, storage site. I mean, again, like with the hydrogen storage sites, for you now, for example, the salt caverns, there nothing really published on microbial rates. Unfortunately, for the porous media, for example, you can, like I showed you the underground sun project. There you have some rates actually, and they saw it also occurring in the field. But then again, there was maybe also other factors in the field. It's sometimes a little bit difficult to really pinpoint everything, to say, okay, this is now only microbiology, but maybe also a little bit diffusion, and maybe also a little bit more mineral reactions, and so everything will occur at the same time. So I'm not aware of a lot of quantitative data yet, but now with more storage projects coming up, I'm sure that we will get more and more. So if you follow, I don't know, the hydrogen storage projects also in the EU, there are now several starting, and several of them have microbiology work packages. So
Okay.
I think we will know more in 2 years latest.
Fantastic. Good. I think we have time for 2 questions. So I'm going to pick this one from Entela. So also thank you for your inspiring talk. Has it been observed that there's any mineral precipitation effect from microbial activity, which at a reservoir scale changes the permeability?
Yeah, yes. This is now a little bit off hydrogen. But for example, when you inject nitrate, as they do for souring treatment, and you have a nitrite reducer, and the first product will be nitrite. And nitrite is actually very reactive for dissolved iron. So you will get actually oxidation of your iron in the water, and then you will have iron precipitation. And this can actually plug your pores. So this is one. And also there has been calcite, microbial calcite formation is known, and it can happen also in the subsurface. And you can actually, it's called biocementation, which is already, I think, very good name that you can actually cement your sand columns. So if you Google for this word biocementation, you will find quite fascinating column experiments where people actually needed to open their columns with hammers because everything was calcified by the microbes. So yes, this is actually happening with different amount of severity.
Thank you. And this is a really nice segue into the final questions that I'm taking here from that plenty of more questions. Apologies to the audience of not taking all of them. So Saida Alameri says, thank you for your interesting presentation. As a reservoir engineer, what would be the negative impact that microbes can cause? Is it just the reservoir souring? Probably more. You sort of partially answered that question already. I guess when you said biocementation could be the loss of permeability, could be other issues. So what are the, as a reservoir engineer, what are the things I need to worry about?
Yeah, so like the plugging for sure. So you can have mineral precipitation can plug. You can have biofilm formation, as I hopefully showed you in the pictures. If you have a lot of biofilm, you can plug very good your pores. Then of course you can have other production, for example, microbial fermenters. They can produce small organic acids, and these acids can actually dissolve minerals. So you can actually have the opposite way. So you can have a dissolution of, for example, carbonates in your reservoir, which can increase the permeability, but also then change the chemistry. You can have, so the pH changes. You can have other mineral interactions, production, of course, of methane. Yeah, so I think this is, and the microbial corrosion, but this is then more topside. This is then less the subsurface. Yeah. So it's a variety of different effects they can have. Again, depending very much on your conditions, depending very much on your reservoir. So it's always, you need to look at your specific field to answer the questions. Yeah.
Great. Thank you very, very much, Nicole, for an excellent talk that certainly for me as a reservoir engineer and geoscientist has opened a lot of new ways, new topics that I need to think about. Thank you to our audience for great questions. Apologies for not taking all of them. And again, thank you, Nicole, for taking the time to answer the questions. And Hardy, over to you for some last comments.
Yes, thank you very much, everyone. I'd like to take the chance, introduce our next week speaker. Will be Dr. Kat Reynolds from Drax Group in UK. Kat will give a talk about negative emissions technologies and the voluntary carbon market. So until next week, 20th of January, 4 p.m. Central European, 3 p.m. British time, 7 a.m. California. Stay happy, healthy, and also tuned in to this channel of geoscience and geoenergy. And we wish you all to see you all next week, and wish you a very good week ahead and a good weekend ahead as well. Thanks a lot, Nicole. It was a great talk and very useful and very informative. Yeah, one. Thanks very much.
Thank you, everyone.
Thank you for the invitation.
Thank you, everyone. Sure. Pleasure.